I've had a couple of people in the past ask me if I'm doing gels with the ancient DNA that I'm working on for my dissertation. No, I'm not doing gels. I have done this though. This is the kayak cell, and it's like fancy gels. It gives us a little bit more data. It can tell us, usually, hopefully, how long in terms of base pairs the actual DNA fragments are. It hasn't been doing that lately. We don't really know why. We're working on that. But fancy gels. We are doing a little pipetting today, but before I do that, I need to label my tubes. So I am going to label the tube with the name of the archaeological site that the samples are from. Right. So now it's ready. And I'm doing this while I'm waiting for, these are our samples. And because we have to keep them in a freezer if we're not working on them, we need to let them thaw out. So sometimes we just hold them in our hands, although my hands are always cold, and the modern DNA lab that I'm working in right now is usually really cold too, and it's cold outside, so yeah, gotta thaw that out. This is labeled. Get back in there. Yep, so I'm gonna thaw this out and then pipette a little bit to mix up the sample. And then I'm going to move 60 microliters from this into the tube that I just labeled. And I'll explain why I'm going to do that in a minute. So I am taking liquid, these are DNA samples, out of this tube and putting it in another one so I can take this tube over to the genomics core of my university. And they are going to run the sample on a tape station. That's going to tell us in each of our samples how many base pairs are the fragments of DNA. And this, we have to do this with ancient DNA because ancient DNA is very fragmented. So we've done this a couple times before, and we're looking at fragments of, you know, at least 100 base pairs in size, but anything that's like a really long number or a big number, like 700 base pairs in size, we're like, that doesn't seem like it's ancient. That's probably some modern contamination. And we do have a couple samples like that, unfortunately. It just happens. That's the nature of working with ancient DNA. Yep, so I'm gonna do that. And we're doing this in preparation to send this out for sequencing, where we'll actually read what the base pairs and in what order they are, and do some analysis with that. And that is what I am working on for my PhD dissertation. There's still some ice in here, so I'm gonna keep holding on to this. I always forget that I need to thaw my stuff out, and then it can take quite a while, especially in the ancient lab when I have a lot of little tubes I need to thaw out. I always forget. Always.